Hey guys, Professor Pinzoni here. So this is lecture 14, and to be completely honest, um, I don't think this is gonna adequately teach you guys methods in how we actually classify and quantify species in forest ecosystems. I had this whole um, plan where we would actually go out into Letchworth Woods and conduct some of these survey methods. But unfortunately, with COVID-19, we can't get out in the forest. And I honestly don't think that me sitting behind a computer screen and, talking to you guys about these survey methods is going to do much. I think I could introduce these topics, but just so you know, I'm going to let you know what is important um, for our upcoming test. With that being said, today we're going to have a brief introduction into how we classify these forest communities. We'll look into why we sample and the things that we do sample. We'll then look into methods to sample forests. We'll look into methods to survey forests. And then we'll specifically look at how forests are classified. And then finally, we'll end up talking about some Western New York examples. So why sample in the first place? Well, for one, forest ecologists, they want to know things like which tree species are located where. How do these species vary in their growth rate? And which animals utilize these different tree species? All very important questions that need to be answered through these methods we will talk about. Another reason why we sample is to take this additional information and accompany them with these satellite images. In reality, they don't really tell us a lot of information. We can't get enough detail from these images to actually quantify the species living there. We can come up with, an area of forest size, but we don't know much else about that specific area via a satellite image. Although we can sort of make out some topography through these images, we can't really tell how tall these different vegetation types are. So we need to utilize forest surveys to calibrate those satellite photos. We need to actually have people on the ground visiting these areas so we know what type of vegetation and the height that corresponds with those vegetation patterns is. We also need to know what is the difference between inventories, samples, and surveys. So this is a key element of this lecture. An inventory consists of a count of all individuals in an area. For example, a count of the number of trees of each species in a park is an inventory. Now, while an inventory is possible to create for small areas, like this small red square in some random place in Africa, it is not a viable option in a large enough area. It would take far too much manpower, far too much time, far too much money to carry out an inventory over a exceedingly large area. A sample contains observations made at one location. Now, a sample may consist of only one observation, for example, the species of the one tallest tree. It can consist of multiple observations, for example, the species of the 5 tallest trees, or it could even be an inventory, for example, the heights of all trees in that location. So there is a slight overlap in these three terms. Now, a survey consists of multiple samples that are strategically distributed across a geographic space. Now, many times, especially in modern forest ecology, really good in-depth studies, they carry out these surveys. Because of their strategic placing, you don't have a large area to actually quantify, but that area, because it's strategically placed and because there's multiples, you actually get a relatively good data set that you can actually analyze, and you could relatively have a lot of confidence in actually better understanding that system you're carrying out that survey in. Some things to sample to understand the forest and its environment. Sessile species. You should know this is a main part of our course here, is studying trees. Trees do not move. Because they don't move, we know exactly their growth pattern has an interaction with that specific area. So sessile species are a great thing to sample to get a better understanding of that local area. In studying sessile species, you can study living species. There's a ton of information in living trees. Their structural characteristics. For example, their height, diameter at breast height, and size of their cavities are all really great information that you can take from living trees. In dead trees, there's still a lot of information there. The species, for example, many times can still be qualified. Their size too. You can still take a DBH a lot of times from a dead tree. You can also look at their tree rings too. I mean, another really useful use of dendrochronology, we use dead trees. Sometimes fossilized petrified trees can give us a lot of information about ancient ecosystems. For vagal species, organisms that are free living, that aren't attached to the ground, you need direct observation or indirect evidence. So for direct observation, it's easiest if the organism you're looking to quantify or qualify are large, are colorful, and are gregarious, which means they congregate in large numbers. And usually when they are gregarious like that, they are more unafraid of humans. So they're not going to just run away, scurry away at the sight of a human. For animals that are small, camouflage, or shy, you're going to have to use indirect evidence to sample them. These forms of evidence come in the form of tracks. Like on the bottom right picture now, we have the differences in a black bear track versus a grizzly bear track. Besides just the size of the paw, you have this different paw geometry. Nests are a fantastic example of this indirect evidence. Now, I'll get into this later on in the lecture, but in 2016, I actually worked the entire summer in the southern Appalachian Mountains, where I worked on this vegetation survey for Dr. Rob Warren. And I basically lived in this cabin for 3 months. It was a really nice cabin, had running water, but it was this ecological station, and there were all these different projects going on. And it was honestly one of the best times of my life. People were coming in and out of this. Some people were staying there for 6 months at a time, some people just the weekend, and everyone was conducting all their ecological research. There was this group of girls that were actually studying the bird species in the area. And mind you, this is some of the most biodiverse places on the eastern seaboard. And these girls were not only amazing at identifying birds hands-on, but they can go through forests and find different bird nests, and not only just where the nesting site was, but they would take a look and look at what the birds were making their nests out of. They would look at nest material. They would be able to identify the eggs in the nest, if there were eggs in there. And it was just an amazing example of using indirect evidence to quantify and qualify the species in an area. I mean, these birds, some of these birds are extremely hard to see and capture hands-on, so you can only use indirect evidence like that. And then finally, we have feces. You can utilize and see and identify the feces of different animal. And to dive a little deeper, you can actually see what those organisms have eaten sometimes. There are clear environmental factors that influence forest growth. Soil conditions such as soil texture and cation exchange capacity. These are terms that we should really understand by now, right? Soil texture and the cation exchange capacity. Canopy cover changes the amount of incoming solar radiation. This is different than leaf area index, which was calculated for an individual tree. Canopy cover is calculated over a larger area that contains multiple trees. So you need to determine this by taking a picture, usually using a fisheye lens. Fisheye lenses are awesome. Then using an image analysis to determine the percent of the canopy through which sky is visible. So you take your focal length of your lens, you can put it, you can analyze the area at the canopy and come out with a value just using a fisheye lens and a camera. So although we learned about how you can sample different animals and different environmental factors that influence forest growth, we're going to focus on these sessile organisms, these organisms that we've spent the most time on, tree species as well as these forest structures. So we're going to focus on the sampling of these sessile organisms. So a structural feature, such as the number of stems of each species in a size class, does vary. So when we look at a forest, you can see there's different size trees. So a sapling is usually going to have a diameter of 1 inch to 5 inches. A pole tree is going to be from 5 inches to 15 inch. A saw tree is from 15 to 30 inches. A DBH of greater than 30 inches is usually going to be an old growth individual. So again, this varies for whatever stand you're looking at. I think it was last lecture or maybe the lecture before that, I brought up an example of an individual tree that was less than 10 inches in diameter at breast height that was over 100 years old. So keep that in mind that these aren't definitive, but for our cases here, this is how we will look at these 4 different classes of trees. Past these structural features, we can look at species features, such as the relative abundance of trees of each species. For example, if a plot of forest contains 5 red oak and 5 sugar maple trees, you could say that the forest was 50% red oak and 50% sugar maple. However, that calculation doesn't take into account the relative size of the 10 different trees. Maybe all 5 sugar maple trees had a very large basal area. Remember, this is lecture 1, I believe, that the basal area of a tree is the tree's trunk, basically a cross section of the tree's trunk, right? So taking a look at the area of the cross section of a tree's trunk will give you the basal area. So you take that value of a single individual and you add it with all the individuals with the same species. That gives you the basal area for, let's say, these maples or oak trees. So these species features can also be expressed as importance values of each species. This is calculated for species A as a percent of all stems that are of species A plus the percent of total stem basal area of the species that is of species A divided by 2. Both of those variables are used in this metric as both are important. Basal area, guys, it indicates the current dominance. A species that has a large basal area is dominating that area. However, if you compare that to the number, so let's say species A has a large basal area, but species B has a large stem number. Stem number represents potential future dominance. So I really like that idea. Basal area indicates the current dominance, but stem number represents potential future dominance. So to continue this early example of a forest that contained 5 sugar maples and 5 red oaks, perhaps the 5 sugar maples were all old growth size, and the 5 red oak were all pole sized. So the basal area of the sugar maple might sum up to be 4,000 centimeters squared, while the red oak, those small individuals, their basal area makes up only 400 centimeters squared, making the percent of basal area 91% for sugar maple and just 9% for red oak. The importance value for the two species would thus be 70.5% for sugar maple, and that would be calculated as 50% because, again, there's 5 individuals of sugar maples and 5 individuals of red oak. So 50% plus 91%, which is made up of that basal area, divided by 2 gives you that 70.5% importance value. For the red oak, it will be 29.5% because 50% plus 9%, that basal area again is 9%, divided by 2 will give you that 29.5% value. In the second part of the lecture, we're going to focus on methods to sample forests. So let's take an unknown area. Let's say you wanted to study some random place in Argentina. This is completely unknown to you, and you really only have photographs provided by Google Maps. Now, there's only so much information, and in modern days, we actually have more information available to us via Google Street View, for example. So I can actually see some forest structure there. If I knew a lot of South American tree species in this area, if I had some background, maybe I can identify some species, but still, you're only going to get individuals that grow well near roads, right? So these data will be biased, but there's more information than there used to be using these resources. But anyway, it's just we know that there's just not enough information utilizing these resources. So regardless of whether you want to determine the relative abundance of tree species, the presence of animal species, or the soil conditions, you can't do a complete inventory of the whole area because that's just going to take way too long. You only have time to study small parts of the area, right? So how do you look at this system in these small samples and have enough confidence in realizing what those systems are actually made up of? Well, we're going to look at 3 types of samples that can be used. We'll look at quadrat, transects, and distance sampling. And this is where I get a little bit sad because we were actually going to go back into Letchworth Woods and use it as a model to actually carry out many of these sampling techniques. We're going to get our hands dirty again, and honestly, I don't know how effective this lecture is going to go. This is something that if you really do need hands-on experience to fully understand how these sampling techniques work, but I'm going to walk you through them anyway because this is how we are doing things at this time. So for the quadrat sample, we need to know what a quadrat is. It is a fixed-sized, square-shaped sampling frame that is placed in the forest. It may be a solid frame for small quadrats or made of flexible tape for large quadrats. Now, small quadrats are used to quantify and qualify seedlings and herbs, these non-woody herbaceous plants, because there is many of these small plants per meter. So again, back to that job I had back in 2016 or 2017. It wasn't that long ago. I think it was 2016 in the Southern Appalachian Mountains. I carried out these vegetation surveys using 1 meter by 1 meter quadrat sampling. Now, our first day on the job, we would put this quadrat down, and you have to literally identify all of the plant species in that frame. First day on the job, we felt so dumb. Me and my partner, Mike, we felt like we knew 3 species. It took us about, I would say, a month of heavy-duty research that we could actually knock out these frames in a relatively short time. But I remember the first 2 weeks, we would be out in the field for 3 hours, 4 hours, and we would have like 1 meter frame done. But as time progressed, we got much better at it, and it was really cool. And by the end of my 3-month research job I was relatively good at identifying these underlying plants, these understory plants. Other studies might want to quantify larger individuals, and they're going to use larger quadrats, maybe a 10 by 10 meter plot made up of flexible tape, because you can't take a huge frame into a forest. The size changes of your quadrat depending on what you're actually studying. Now, note that a quadrat is different than a quadrant, right? These are 2 different words. So a quadrat is a frame with 4 sides that is used to, in our case, to study these vegetation patterns. A quadrant with an N is one part of a shape, any shape that is divided into 4 parts, right? So it's a shape that's divided into 4 equal parts. Now we are going to use quadrants in distance sampling in section 2D coming up, but not in quadrat sampling. A transect sample utilizes a straight line that runs through an area of interest, such as a forest. The line is then divided into sections with a regular length, usually around 10 meters long. With each section, the total fraction of the line that is covered by the crowns of different species of trees is measured. Now, this is relatively easy to place the line transect in forests. However, there are 2 limitations to this approach. The first one being that it can be very difficult to recognize species of trees by leaves in the canopy. Really tall, mature trees, their crown is usually much higher. I find myself really struggling sometimes with really old trees that don't have any low-lying branches. I try finding, leaf debris on the ground. But you guys know, as a tree matures, its bark changes through time, and its bark might not be as easy to identify. And then you have its leaves hundreds of feet over your head that you're using binoculars to try to distinguish. So species recognition is a downside of this approach. Second, it is really tiring to constantly being looking up in a tree crown. I mean, you're looking directly up. This does get tiresome when you're doing it thousands upon thousands of time during this type of transect sampling. A belt transect sample has two parallel lines instead of just one line in a regular transect sample. These two lines are then divided into sections, and these sections are again around 10 meters long. Then, in each section of the belt transect, all individual trees are identified to species and counted, and their relative abundance is calculated. For example, 30% sugar maple in this transect, in this section of the transect. So it is going to be harder to place two parallel lines compared to just the single line transect sampling method, but it is easier to record individual trees than it is to estimate canopy cover in the previous sampling method. So all in all, both of these sampling methods that use transects, they are extremely useful in recording how the relative abundance of tree species changes along an environmental gradient. So in this picture in the bottom right, this is obviously the elevation is changing. You're going to have different light conditions, different moisture conditions, different wind conditions because of this increase in elevation. So having a line transect and seeing the species and the number of individuals and the size of individuals change up this transect is extremely useful. So not only in elevation, but with places that vary in soil moisture, with distance away from a lake, that is also a great use of a transect method. Researchers using distance sampling, they're going to measure the distance from a center point to the nearest tree. And then they're going to record information of interest about that tree, such as species or diameter at breast height. So the distance could just be measured to the one closest tree or the closest tree in each of the four compass quadrants. Again, this is quadrant with an N, right? A quadrant is different than that frame quadrat. You can identify and record data using this quadrant method, recording the closest tree in each of the cardinal quadrants. So you're going to have the closest tree in the northeast, the northwest, the southwest, and the southeast quadrant. The use of quadrants allows a more reliable local estimate of forest density than is provided by the use of just the one closest tree. So again, forest density is the number of trees per acre. So using the 4-quadrant method, you're going to be closer to the actual forest density. Let's talk about the number of samples that one should use. The more samples in a survey, the better, because they're going to provide more information on the spatial and environmental differences within the study area. However, there's time limitations, right? And with time limitations, you come with financial limitations. Because people, they need to get paid. So time limitations usually decide the number of samples. 30 samples is a typical minimum number in a local forest survey, but thousands are required if a large or complex area is studied. So, for instance, a grassland ecosystem is going to require less sampling usually. There are some very biodiverse grasslands, but more arid grasslands with only 10 to 20 species They don't need a lot of sampling. However, if you go to a rainforest near the equator, tropical rainforest, you're going to need a lot more samples than just that minimum 30 that I just described. In this next part of the lecture, part 3, we're going to be focusing on the different methods of surveying forests. So this is different than sampling forests, as a survey actually contains samples that are strategically distributed throughout the study area. So surveys, again, they are more comprehensive than just singular sampling. They actually utilize those sampling techniques that we just learned about. We're going to look at 2 ways surveys can be spatially biased and 3 ways in which surveys can spatially distribute samples in a non-biased manner. So first, let's focus on these biased surveys. So if you go on this path, this road right here, and do your sampling on this nice path, number one, it's going to be easier, right? And because of that, there's going to be more surveyors that are, that have a tendency to stay on a specific path, but this is extremely biased, right? Even though this road offers easy access into a forest, sampling along them would result in extremely biased data about the forest, as a path and road are typically going to be located on a dry valley bottom and avoid those wet areas, right? In addition, there's going to be more available light because there is this canopy openness over that road. So you're going to have individuals and different species that are going to require more light and be adapted to less water. So already your research is looking quite biased. Now, wandering through a forest is also biased because you might think you are wandering randomly, but you will be unconsciously drawn to certain things and avoid others. So this, by you being influenced by obstacles and terrain difficulty, you're unconsciously going to have a biased data set. Biased surveys will therefore result in certain types of vegetation being oversampled and other types of vegetation being undersampled. So to describe some non-biased surveys, I'm going to utilize Letchworth Woods right on campus as an example. So the first non-biased survey we're going to look at is completely random. So in a random non-biased survey, you're going to have quadrant samples, point samples, or transect samples that are randomly distributed through a forest. A random number generator is used to determine the geographic locations of the random points or quadrants or the beginning of the random transects. You're then going to use a GPS device to reach the point or quadrant and a compass to orient the transect. Now, this approach is simple but can miss uncommon but important forest types. So that is a downside. It can miss uncommon but important forest types just because of its complete randomness. So a systematic non-biased survey is going to utilize quadrants, points, or transects that are distributed in a regular manner. For example, a number of line transects may run in an east to west direction, parallel to each other and separated by 100 meters. Look at this figure to the right now. You have these lines that have this systematic orientation. This approach is simple to implement, but it has its shortcomings as well. Similar to the random non-biased surveys, this systematic survey can still miss uncommon but important forest types. The third and final non-biased survey method that we're going to learn about is the stratified random method. Now, the stratified random method, you're going to need access to a satellite image prior to your sampling. So first is first, the site is going to be divided into different strata or groups using these satellite photos. So looking at these images, you're going to actually select areas that are visually different. So let's take a look at this picture in the bottom right, and we can actually see these darker, different looking areas. Then the next thing we're going to do is delineate the forest into interior portions and edge portions, because usually the outer 100 meters of a forest is more influenced by wind, dust, and salt and other influences. So this blue region is going to be the forest interior for our Letchworth Woods. Next, we're going to divide further into areas with expected differences. So if it's, a terrain that varies in elevation, you're going to try to encompass those elevation differences. Here, we're going to use aspects. So we're going to use a compass, these cardinal directions, to further divide these areas. Then randomly distributed points, quadrants, or transects are going to be used in a combination of these visual differences plus these compass aspects plus the edge or interior. So you guys see we have points in each of these compass aspects. We have points in the interior and exterior, and we have points throughout each of these features. This is definitely the most complex approach to implement, but it is most likely the best and greatest approach to visit and record the full range of forest types in a non-biased manner. In this next portion of this lecture, we're going to look at how maps are created and how we classify these forests. The relative abundance of each species in each sample can be used to create maps. So a map can be created of the relative abundance of a single species, but tens or even hundreds of species might be represented in the sample area. If maps were created of each species, then there would be too many maps to deal with. So the solution is to create classes that contain samples that are similar to each other and different from other samples. So we are going to look at 4 different ways to group the samples into classes. In the first 2 methods that we'll learn about, dominant ordinals and dominant sums, this table to the right is utilized. So for dominant ordinals, the ordinals are created by ranking species by their relative abundance. First most abundant, second most abundant, third, and etc. So each sample could then be named using the one most abundant species in each sample. Using the sample data, we get the names for plot 1 as A, plot 2 we would call plot 2 A, plot 3 would be B, and plot 4 would be B. So you'll notice that the letter I assigned to each plot was the most abundant species. Alternatively, we could assign the name to each sample by the first and second most abundant species together. So using this sample data, we now have the names for plot 1 as AC, as those are the 2 most abundant species. For plot 2, it would be A and D. For plot 3, it would be B and E. And for plot 4, it would also be B and E. So that is how we can classify these forests utilizing these dominant ordinals. Dominant sums are used to take into account not just what species is most abundant, but how much more abundant they are than the remaining species. The name class must represent top species that together exceed some critical value. So for example, if 50% is that critical value, one species may dominate and it only gets one name. In other samples in which a number of species dominate, then it will have 2, 3, or even more names. The second species included would be the second most abundant species in the plot, and the third species included would be the third most abundant species in this plot. Using the sample data, we now get the names A as the critical, as that critical value is reached in species A for plot 1. In plot 2, the species would be AD, as both of their percentages would exceed that 50%. And then in plot 3, it would just be B. In plot 4, you would actually have B and E, as both of those values would exceed that 50%, that critical value. Next, we have statistical clustering. In statistical clustering, this method does not just employ information on differences in most abundant species, but also uses information on uncommon species and on species that are commonly co-occurring. The degree of similarity between all plots is calculated, and statistical methods are then used to group them together in a specific number of classes. The statistical method for doing this are too complex to really even get into, but we have seen more use of this in more recent times. The final method of forest classification is the use of standardized classification. So in the 3 previous methods, ordinals, sums, and statistics, a forest could result, a part of the earth could result in forests that contain the same tree species, but given a different name based on the geographic area. So this leads to, for example, 3 states creating slightly different classes of vegetation that did not align well at the state borders. So cartographers call this an edge matching error. As you can see, we have Oregon, Idaho, and Nevada here, and they have totally different vegetation patterns, even though they're just separated by this man-made state line. So by using a standardized approach, by actually having standardized names for these different vegetation types, you can actually map out a better representation of the vegetation. So a solution to this is the use of the standardized quantitative rules to determine what vegetation class of those samples will be placed. So here is an example of these standardized classification system. So S is defined by an open community by 25% canopy cover of trees. The dominant species are shrubs, herbs, or cryptogamic plants, mosses, lichen, etc. So we call that an open upland. So there are several of these different classifications, and this standardization made it much easier for complying states carrying out these vegetation surveys. Now, the United States actually uses its own classification system, the National Vegetation Classification System, also known as NVCS, and this system has 5 top levels that are based on forest structure. So for example, forest uplands versus barrens, those 2 terms describe forest structure. Then its lower levels are based on the actual species composition. So by using these 2 levels, the top and the lower levels, you can come out with a standardized term for that particular plot that you're looking at. In the United States, we have over 6,000 recognized vegetation types. It's actually 6,161 different vegetation types to be exact. Almost 250 of those are in New York State, 247 to be exact, and some of them are actually critically imperiled. So we have some very rare plant communities even in New York State. For example, we have a maritime holly forest found on the back side of sand dunes in New York State and New Jersey. These are dominated by small but really old holly trees. Holly trees are these evergreen shrub-like plants that are up to 30 feet tall. Some of them are 300 years in age, and there's a subdominant tree of sassafras and smaller black oak. So the data collected at any one sample location can be classified using the NVCS system, this system that our great country has come up with. If a series of samples were distributed using a survey and they were all classified using the NVCS, then a map of the vegetation classes for that surveyed area could then be made. So here is an example of these utilizing this system and these maps. Each color represents a different vegetation system. I really like looking at these maps. You can see the Sierra Nevada north of where these colored sections actually start, and you can see behind them, right to the east, you have this very arid system because, again, from the last lectures, you have that condensation occurring as elevation increases, so that water gets wicked away from that air, and you have that drier and hotter parcel of air on that eastern side past that the Sierra Nevada mountain range. The final part of this lecture, we're going to look at Western New York examples of these surveys. So 2 surveys of forests in Western New York will exemplify the different survey and sampling methods. So systematic and distance methods were used by the Holland Land Company in 1798 when they surveyed Western New York lands to facilitate Euro-American settlement. So they had sample locations systematically distributed every half mile along edges of townships that were typically 6 by 6 miles in size. These survey lines later became roads like Niagara Falls Boulevard and Transit Road. At every sample location, they divided space into 4 quadrants. Then recorded the species of individual tree and each of the 4 quadrants that was closest to that sample location. So there were thousands of trees in total. The stratified plus random approach is taken by the U.S. Forest Service Forest Inventory Analysis. So FIA represents that Forest Inventory Analysis. Now this was a survey across the United States. A sample location is allocated for each 6,000 acres of existing forest, which stratifies the space geographically. A sample is placed randomly within that 6,000 acre area and then is revisited every 5 years. They record information on which tree species are there, their size, and their growth rate. So from those 2 surveys, we actually have these 2 single species maps created from 1790 and the 1990s. You can see using these data how species change through time, interacting with human development. So in 1790, Western New York was completely dominated by beech forests. Nowadays, they're the most limited species that you see. Sugar maple seems to be doing rather well here. We have poplar trees becoming more and more prevalent in recent time, and ash also became more and more prevalent now. The emerald ash borer, an invasive insect, has decimated these ash species. So if we had a graph that showed the 2000s, you would again see a different vegetation map, especially for ash. Now, for poplar, these trees really like disturbance. They're some of the first successional species, and they're showing up a lot more in Western New York in more recent times because there are a lot of human disturbances. Because we remove forests, we change our land use, and as we abandon those lands, usually one of the first tree species that shows up are these poplar trees. Here we have maps of 4 different forest community types in Western New York. Now, these communities were obtained through a statistical clustering approach. The horizontal lines show where that community type was present in 1790s. So again, horizontal defines the 1790s, while the vertical line shows these species types in the 1990s. These maps show not only did the individual species change in abundance, but so did the overall abundance of different forest communities. In 1790, most of Western New York was dominated by the beech sugar maple forest class. In the 1990s, that forest class was no longer present, and it was replaced by 3 other forest classes that had only limited coverage in the 1790s. So that's a really interesting idea. That beech sugar maple forest type that was dominant in the 1790s isn't even a forest class of present day, right? So forests are constantly changing, especially due to human interaction. Hopefully, this lecture has helped us better understand how we classify and how we actually hands-on get into these forests and can extrapolate and get data from those lands to better understand how those systems change through time. So yes, thank you for your attention today. We have an exam on Thursday, the 9th. I'm going to post that early in the morning, and it's going to be the same setup. 25 multiple choice questions, and you'll have a choice to write 1 essay. I'll give you 3 essay questions. I hope you guys are staying happy and healthy and are learning something from this. Have a great rest of your day, and I will catch up with you next week. Got some really exciting stuff next week. I went to Zor Valley yesterday to take some footage for next week's lecture, so there is that to look forward to. Again, have a good day and keep learning and stay healthy.